I would firstly like to thank the organizers for giving me the opportunity to give a talk over here. I'm sorry about the initial glitch. I didn't realize that my recorded audio will start playing. Hi, I'm Niladri. I work in the lab of Professor Christine Orango at UCL. Today, I'll be talking about characterizing and explaining the impact of disease-associated mutations in proteins without known structures or homologues. This work was done in collaboration with David Baker at University of Washington and Samir Velankar at EBI. Uh, to as so the main objectives of the study, as it must be evident from the talk title, is to model human organisms that do not have any close homologues in the PDB. And why would we want to do that? We would want to do that because we wanted to use these models to explain disease associated mutations, which could be either based on proximity to functional sites, uh, structural destabilization, or pathogenicity. For this, we created a data set of that we obtained from VAR site, which contains some 553 proteins, and the corresponding disease-associated mutations, we got it from Hamzavar, which were a total of 1730 mutations. When we started this work in collaboration with David Baker, we were using Gremlin, which was the then tool, state-of-art tool. But as we all know, AlphaFold came soon after that in CAS 2020, and which used a deep learning model to predict protein structures. And as it can be seen from the GDD scores on the plot, AlphaFold 2 outperformed all other techniques by a lot. Soon after that, David Baker's lab came up with a tool called Rosetta Fold, which was also based on similar techniques of deep learning, and we hence used both AlphaFold and Rosetta Fold for our work. Uh, uh, Later in the year, AlphaFold released the models of 21 model organisms, which also contained the human models that we were trying to build and model. So we decided to go ahead and use the AlphaFold models from the AlphaFold EBI database, and with the collaboration of David Baker, we thought that we could also model the proteins using Rosetta Fold. So these are human proteins which could be multi-domain, and as a result of it, we could have the domains built very well in the protein structure, but the orientation of the domains might not be very well. So as a result of which, we chop the protein up into the individual domains using a technique called cath resolve hits, which was published in Bioinformatics in 2019. So what it does, it takes the protein sequence and it aligns it against HMMs of cath families or PFAM families, and it gets protein sequence domains out of it. So for our task, what we did first was we assigned it to cath domains, and whatever we couldn't assign to cath domains, we assigned it to PFAM domains, and whatever we couldn't assign to cath or PFAM at this moment, we call them new FAMs. So for our study in our data set, we had 553 proteins that I had already said from VAR side. Oh, for those proteins, we got some 309 CAD domains, some 416 PFAM domains, and the remaining around 500 domain-like regions, which we couldn't assign to CAT or PFAM. We saw that the CAD domains are modeled better. So these are the three histogram shows for each type of domain. What is the PLDTT score for each domain, which is the average of the LDTT score of each residue. Uh, and the vertical line over there is a 70 cutoff that is suggested by them, above which it model is considered well. We see around 90% of the, much over 95% of the CAD domains were well modeled. Around 80% of the PFAM domains were well modeled, while only less than 50% of the PFAM domains were well modeled. So we wanted to check what could be the reason behind this. So what we checked was, we tried to see if there was a difference in the alignment diversity in each of the domain types. We didn't have access to the AlphaFold multiple sequence alignment. However, we had access to the Rosetta Fold ones. So we used the Rosetta Fold multiple sequence alignment to check it. Over here is a density plot of the NEF values. So NEF is the number of sequences in the multiple sequence alignment at an 80% cutoff. And in red is the cat domain, in green are the NEF values for the PFAM domain, and in blue are the NEF values for the new FAM domains. We see that the cat domains have much higher NEF, while the new FAMs have a much lower NEF value, indicating that the alignment diversity for those regions of the proteins were quite low, while the alignment diversity of the cat domains was quite high. So, however, we couldn't find any clear correlation between the information content of the MSA as we calculated from NEF, which is shown by the dot plot, along with the model quality. So we also tried to check along with the diversity of the multiple sequence alignment if we had other characteristics which were different in the different domains. We next checked the disorder percentage in the domains. And we used IUPRED2 for the purpose. And over here again in red is the percentage disorder of the sequence in cat domain, green PFAM domains, and in black dotted lines are the new FAMs. And it's very evident that the cat domains are well structured domains with very low percent disorders, while the new FAMs are not quite well structured. So as a result of which, if a domain is well structured, it will have good LDT score, and yeah. And we also noticed that the quality of the model reduced with increase in the percentage LDT score. Over there is the dot plot, where along the x-axis we have the percentage predicted disorder. Along the y-axis in red dots are the AlphaFold scores, blue dots are the Rosetta Fold scores, and there is a negative correlation of the percentage disorder with the score of the models. We then compared how the AlphaFold models with the Rosetta Fold models. So over here are the plots for, along the x-axis is the Rosetta Fold model quality score, y-axis is the AlphaFold model quality score. A larger number of the AlphaFold 2 models were better modeled as compared to Rosetta Fold. And we see that a larger number of cat domains were better modeled. However, if you look at the plot, you will see that there were certain domains of Rosetta Fold which were of good quality, where the AlphaFold models were not of good quality. So as a result, using Rosetta Fold not only gave us additional confidence in the models models, also it gave us other models where AlphaFold model quality was not good to be used in our analysis. We also structurally compared the AlphaFold 2 and Rosetta Fold models. For this purpose, we used GZAM to superimpose the structures. Again, at the RMSD scores for the 3 different domain types. And we see that a larger percentage of cat domains have a low RMSD, meaning that both AlphaFold and Rosetta Fold say that the structures are very similar to each other. And new FAMs has a higher percentage of domains which had higher RMSD, indicating that the two modeling tools didn't predict a similar structure for it. And as expected, the model quality also reduced with an increase in RMSD. So again, in the first column is the CATs, second column PFAM, third column new FAMs. We see that as the RMSD of the model increased, the model quality went on decreasing. So after we had all these models, we went to the next task of utilizing these models to explain disease-associated mutations. And we used different tools for predicting the functionally important sites. We predicted the conserved sites using score cons, ligand binding sites using P2 rank, and interfaces using MetaPPISP. And in our data set, we had around 1730 disease-associated mutations, out of which some 1,300 mutations were in the high-quality domains of AlphaFold or Rosetta Fold, and even those residues were of high quality because we could only use data if both the domain and the quality of individual residue was of high quality. Out of the 1,300 mutations, around 800 mutations were in or near a predicted functional site. We also did some analysis with destabilization and pathogenicity, which I will come back to later in my slides. So there were multiple evidences where the same mutation was predicted as a functional site based on multiple tools. So in case a site is predicted as an interface and also as a conserved residue, we would have higher confidence in that site being actually a functionally important site. So around 44% of the mutations were predicted to be on or near a functional site based on more than one predictors. Here, this is a Venn diagram that shows the overlap between the different predictions. So I will show you a few examples as to where we could explain some mutations based on proximity. So the first one is a structure of putative sodium couple neutral amino acid transporter 8. The ligand binding site is in blue, and the disease-associated pathogenic mutations are in red, as evident that the mutations are in the predicted ligand binding site. Similarly, we have two other examples where we have used an AlphaFold and a Rosetta Fold model to predict the functional site and showed that the mutations were on or near a predicted functional site. Along with this, we also check the structural stabilization of the mutations. Uh, uh, we use two tools for the purpose because delta delta G calculations are a bit tricky, and we didn't want to base our final conclusions on only one tool. So over here at the delta delta G for fold-X on the x-axis and Dynamute2 on the y-axis, we do not quite see a clear correlation between the delta delta G, but however, we assume that if two tools are saying that the mutation is structurally destabilizing, we assume that it is it should be structurally destabilizing. So out of the 1,300 disease-associated mutations in our data set, around 700 mutations were predicted as destabilizing by fold-X. Of these, some 272 mutations, they couldn't be explained based on proximity to a functional site. Most probably, they were in a buried region of the protein, and hence they mostly destabilize the structure of the protein and not a functional site. Around 95% of the mutations that were predicted to be destabilizing by fold-X were also predicted destabilizing by Dynamute2, and we have higher confidence on those 95% of mutations to be actually structurally destabilizing. Next, we also check the pathogenicity of the mutations. We used a tool called MutPET2. Over here is the histogram of the pathogenicity scores of MutPET2 for each of the mutations, and the cutoff is 0.61 as suggested by them, above which we consider a mutation as pathogenic, above which they consider the mutation as pathogenic. Around 80% of the disease associated mutations were predicted as pathogenic, and 8 of those pathogenic mutations could not be explained either by proximity to functional site or structural destabilization. So bear in mind, we have used Rosetta Fold models also for all our analysis, and the Rosetta Fold models helped explain more mutations. So there were some 93 disease-associated mutations which we couldn't explain using AlphaFold, either because the AlphaFold model quality was low, or so over here, first let me take you through that figure. The figure is a superimposition of an AlphaFold and a Rosetta Fold model. We see that the two models are very structurally similar, but we have a small change in the loop orientation, because of which the top part of the model are modeled separately. So as a result, if we use the two models, we would get different explanations of functional sites, because the interactions would be a bit different in the two models. So we couldn't explain 93 mutations. We could explain 93 mutations using Rosetta Fold model, primarily because either the corresponding AlphaFold model was of a low quality, or the mutations, it didn't fall within the distance cutoff of the predicted functional site. That means like it just was a bit away from, I mean, say we have a distance cutoff of X angstrom, within which we say it is okay to be there. It was, say, like X plus 0.1 angstrom away in AlphaFold model. In some cases, we also missed the prediction because it didn't meet the cutoff set by the tool to be called as a functional site. Say, like the tool would say it is a functional site at 0.5, while AlphaFold model gave it a thing of 0.4 or something. So we had around 492 disease-associated mutations, where both AlphaFold and Rosetta Fold predicted it as a functional site, and we have higher confidence in those predictions. Along with deleterious mutations or pathogenic ones, we also compared polymorphisms to see how polymorphisms were different from each other, from the pathogenic mutations. In red are the polymorphisms, in blue are the disease-associated mutations. The first plot is of a pathogenicity score. We see that more disease-associated mutations are pathogenic as compared to polymorphism. Second one is the delta-delta G predictions. We see that a larger number of disease-associated mutations have higher delta-delta G of mutation as compared to polymorphism. The third plot below is of the solvent accessibility. We see a larger percentage of the disease-associated mutations were buried, having a low solvent accessible area. If you look at the table, the first column, we see a larger percentage of disease-associated mutations were on or near a predicted functional site as compared to polymorphisms. With this, I come to an end. To summarize, we had around 70% and 50% of the modeled AlphaFold domains, which were of good quality. Around 56% of these domains had an RMSD of less than 2. The CAT domains were modeled better predominantly because the sequence diversity was higher. Also, the CAT domains are lower disordered compared to PFAMs and NEUFAMs. We could explain 79% of the mutations in good quality regions. We had higher confidence in the predictions from models. In case if it had low RMSD between an AlphaFold and Rosetta Fold model, the sites were predicted functionally or structurally destabilizing or pathogenic based on more than one predictor. Also, if both AlphaFold 2 and Rosetta Fold models could explain the disease-associated mutations, the last conclusion is about that the disease-associated mutations are closer to a functional site, more structurally destabilizing, pathogenic, and buried compared to polymorphism. We have published it in briefings in bioinformatics. If you want to get it idea, you could scan the QR code. You could contact me via Twitter, LinkedIn, or an email. You can also contact the lab or get updates about what we do in the lab from that website. To acknowledge, I would like to acknowledge Ian, Nico, and Christine from Cath Lab, Ivan from David Baker's lab, and David Baker and Samir from EBI, and DBSRC for funding. And that's all. I think I do not have time for any questions. If you want, you can meet me at the coffee break.